Welcome, everyone. Good afternoon, and thank you for joining us for the Developments in COVID-19 Therapeutics webinar, hosted by Drexel University Online and presented by a panel of distinguished faculty from the Drexel's College of Medicine. My name is Reina Lopez, and I'll be moderating this presentation. Before we get started, I'd like to take care of a few housekeeping items. First, I'd like to make sure everyone can hear me. You should see a button with the icon of a raised hand. Click on that icon if you can hear me. Okay, great. Lastly, I'd like to let everyone know that today's presentation is being recorded and will be sent in a follow-up email. During today's webinar, our presenters will be speaking with you about the virology of COVID-19, development of the antiviral therapeutics, vaccine development, current therapeutic initiatives, and an overview of clinical trials. They will also answer as many questions as they can at the conclusion of the presentation. Before we begin, I would like to take a quick moment to introduce Drexel University to you. For those of you who aren't familiar, Drexel has a long-standing history in providing quality education. Founded in 1891, Drexel is one of the largest not-for-profit universities in the US. We are located in Philadelphia and have more than 24,000 students. In addition to our diverse portfolio of traditionally on-campus students, we are also proud to offer more than 150 programs online. For questions and comments during the presentation, please use the chat or comment feature. Today's speakers will be presenting live and will answer your questions at the end of the presentation. We will try to get to everyone's questions as best we can. Any questions that we aren't able to answer before the conclusion of today's event will be added to a question and answer webpage. We will provide a link via email after that page is updated with your great questions. Today, we are fortunate to have with us a panel of distinguished speakers from Drexel University's College of Medicine. I would like to introduce to you Dr. Sonia Navas-Martin, Dr. Fred Krebs, Dr. Michele Kutzler, Dr. Paul McGonigle, and Dr. Richard Mangiano. With that, I will hand it off to Dr. Navas-Martin.
Can you hear me now?
Yes, Sonia, we can hear you.
Okay. My name is Sonia Navas-Martin. I am Associate Professor of Microbiology and Immunology at Drexel University College of Medicine. My talk is going to give you a basic overview on coronavirus molecular virology and the implications for therapeutics and vaccines. Next slide, please. Coronaviruses are, can you actually please the next slide? Coronaviruses are the largest group of viruses belonging to the Nidovirales order and infect birds and mammals, including humans, causing diseases with variable mortality that affect lung, the kidney, the gastrointestinal tract, and the central nervous system. The Nidovirales order include other families of viruses, but none of these infect humans, although they can induce massive mortality in other hosts. Next slide, please. Coronaviruses can be transmitted by respiratory droplets with some evidence of fecal-oral transmission for some animal coronaviruses. All the 7 human coronaviruses that we know have been transmitted from animals to people, and this is called zoonosis. According to current evidence, SARS-CoV-2 is primarily transmitted between people through respiratory droplets by direct contact with infected people and indirect contact with surfaces in the immediate environment or with objects used on the infected persons. In the context of SARS-CoV-2, airborne transmission may be possible in specific circumstances and settings in which procedures or support treatments that generate aerosols are performed, and this is extremely important in hospital settings. Next slide, please. The subfamily Coronavirinae can be split into 4 main genera of coronaviruses on the basis of their serological and genomic properties, that is alpha, beta, gamma, and delta coronaviruses. Human coronaviruses belong only within the alpha and beta coronavirus group. Beta coronavirus can be further split into 4 lineages, A, B, C, and D. The recently identified severe acute respiratory syndrome coronavirus 2 is classified under the lineage B of beta coronavirus. Next slide. Bat species are among the oldest mammals and represent about 20% of mammalian diversity. While numerous animals have been surveyed in the past decade, bats continue to be among the most abundant source for novel viral sequences and are likely the major natural reservoirs for alpha and beta coronaviruses. Bat diversity and long coevolutionary relationship with pathogens provide the opportunity for cross-species mixing and maintenance of quasi-species pools of viruses that can infect a range of hosts. On the basis of current sequence databases, all human coronaviruses have animal origins. SARS coronavirus, MERS coronavirus, Netherlands 63 coronavirus, and 229E coronavirus are considered to have originated in bats. Human coronavirus OC43 and human coronavirus Hong Kong 1 likely originate from rodents. Domestic animals may have important roles as intermediate hosts that enable virus transmission from natural hosts to humans. In this regard, for example, coronavirus OC43, a common cold-causing virus, jumped from cattle to humans approximately 120 years ago. The newly identified SARS coronavirus 2 shares 96% whole genome identity with a particular bat coronavirus strain. However, coronaviruses usually pass into intermediate hosts, such as civets, cats, camels, before jumping to humans. And pangolins are currently considering a potential intermediate host for SARS coronavirus 2. Next slide, please. There are 7 human coronaviruses that we know so far. 4 strains are endemic and include 229E, OC43, Netherlands 63, and Hong Kong 1. These endemic strains cause mild respiratory infections and are responsible of about 30% of the human common cold. However, and less frequent, they can also cause more severe lower respiratory infections in immunocompromised patients. These viruses do not induce durable immunity, and there are frequent cycles of infections. In contrast, emerging coronaviruses include highly pathogenic coronaviruses, such as SARS, MERS, and SARS-CoV-2. These coronaviruses infect the lower respiratory tract and cause acute lung injury that can progress to fatal acute respiratory distress syndrome and septic shock with multi-organ failure. It is important to highlight that SARS coronavirus 2 is the name of the virus and COVID-19 is the name of the disease. Next slide. Coronaviruses are envelope viruses that have a round, sometimes pleomorphic virion of approximately 120 nanometers in diameter. The genome RNA is complex with the basic nucleocapsid protein to form a helical capsid found within the viral membrane. The membranes of all coronaviruses contain at least 3 viral proteins. These are the spike, a type 1 glycoprotein that forms the teplomers on the virion surface, giving the virus its corona-like morphology. The membrane of matrix protein and a small membrane protein E that is a highly hydrophobic protein with an anion channel activity. The coronavirus spike protein plays vital roles in viral entry, cell-to-cell spread, and determining tissue tropism. It also contains B and T cell epitopes and is a major target for neutralization and vaccine strategies. Next slide. The positive sense single-stranded RNA genomes of all coronaviruses have a similar structure, and they are the largest genomes of all RNase virus known. The 5' N, approximately 20 to 22 kilobases, carries the replicase gene, which encodes for at least 16 enzymatic activities. The replicase gene products are encoded within two very large open reading frames, 1A and 1B, which are translated into two large polypeptides via frame shift mechanism, involving a pseudoknot structure forming by the genomic RNA. The structural proteins are encoded within the 3' 1/3 of the genome in the order spike, small envelope, matrix, and nucleocapsid. Each group of coronaviruses, in addition, encodes a group of unique small proteins, and some of them can interfere with the host's innate immune response. Next slide. The coronavirus life cycle begins with the binding to cellular receptor, followed by membrane fusion, as well as viral RNA and protein synthesis in the cytoplasm. Coronaviruses can enter the cell through both pH-dependent and independent mechanisms depending on cell type. The polyproteins 1A and 1B are co-translationally processed, resulting in the formation of the replicase complex, a set of nested subgenomic messenger RNAs and genomic RNA, which possess both the same 3' end and a common 5' leader sequence derived from the 5' end of the genome is then transcribed. Virion assembly is achieved by binding into intracellular membranes, and virion release is accomplished through the secretory pathway. A very important characteristic, and in contrast to all RNA viruses, is that coronaviruses encode an exoribonuclease activity called XON that is encoded by the non-structural protein 14 and is distinct from the viral RNA-dependent RNA polymerase that regulates coronavirus replication fidelity. Next slide. The innate antiviral response is the first line of defense against viruses. Type 1 interferon are important antiviral and immunomodulatory agents that result from the activation of the innate immune response. Coronaviruses have multiple mechanisms to evade the innate immune response, most notably with the suppression of type 1 interferon production and signaling pathways. As the first line defense in the immune system, suppression of the innate immune response by these coronaviruses impede their host ability to restrict infection, causing significant disease. SARS coronavirus and MERS coronavirus, as well other coronavirus such as the murine coronavirus, suppress type 1 interferon responses at multiple levels. Having the overall picture of the mechanisms of coronavirus innate immune evasion is key to understand pathogenesis, but also to identify potential therapeutic targets. Next slide. A recent study provides evidence that host cell entry of SARS coronavirus 2 depends on angiotensin converting enzyme 2, a cell molecule that SARS coronavirus identified in 2003 also used as receptor. This recent study also identified that a cellular serine protease called TMP-RSS2 is involved in SARS coronavirus 2 spike protein priming, which entails spike protein cleavage and allows fusion of viral and cellular membranes. At least in cell culture and using a surrogate system of infection, SARS-CoV-2 entry can be blocked by AC2 antibodies and a clinically proven inhibitor of the cellular serine protease TMP-RSS2. Next slide. Next slide, please. Moreover, the same study suggests that antibody responses raised against SARS coronavirus, the previous slide, please. Thank you. The same study suggests that antibody responses raised against SARS coronavirus could at least partially protect against SARS coronavirus 2. The identification of a specific SARS coronavirus neutralizing antibodies is a key area of intense research and have important implications for our understanding of SARS coronavirus transmissibility and pathogenesis, and may actually reveal targets for therapeutic intervention. Next slide. Next slide, please. Thank you. Factors responsible for the unusually high pathogenicity of SARS and MERS coronavirus and the recently identified SARS-CoV-2 are poorly understood. However, insights from a mouse model of the 2003 SARS coronavirus demonstrate that type 1 interferon and inflammatory monocyte-macrophage promote lethal SARS coronavirus infection, and identify type 1 interferon and inflammatory monocyte-macrophages as potential therapeutic targets in patients infected with pathogenic coronaviruses. Next slide. Next slide, please. Although there is a strong evidence that cytokine storms and immunopathology can occur during pathogenic human coronavirus infections, we do not yet have a sufficient understanding of the specific factors responsible for the exuberant inflammatory responses. Studies from human autopsies and animal models strongly suggest a pathogenic role for inflammatory cytokines and chemokines derived from innate immune cells, such as inflammatory monocytes, macrophages, and neutrophils. Therefore, therapeutic interventions targeting these pro-inflammatory cytokines and chemokines could provide beneficial in ameliorating detrimental, exacerbating inflammatory responses. And finally, my last slide, thank you. Finally, our knowledge and further investigations on virus-host interactions, pathogenesis, and animal models is key for the development of antivirals and vaccines. Thank you.
Thank you, Sonia. I am Dr. Fred Krebs. I'm also in the Department of Microbiology and Immunology here at the College of Medicine. I want to thank all of you for joining us today. It's probably unnecessary to point out that it's abundantly clear that there is a critical need for therapeutic drugs effective against the virus at the center of the COVID-19 pandemic. That virus is SARS-CoV-2. However, this urgent need is not at odds with the typically lengthy process of drug development. So my purpose today is to shed some light on the drug development process and how this process is being adapted to fill the needs of the current crisis. Next. The drug development process is typically viewed as a pipeline, a linear process that proceeds from laboratory experiments and work in animal models, progresses into clinical trials, and then ends up with an approved product that shows up in the drugstore. It's available for prescribing by physicians. Next slide, please. This process, again represented on the top of the screen here, is a lengthy process. The process of drug development typically takes 10 to 15 years from the time you start investigating a drug until the time you get to a product that's approved by the FDA. Of course, this process is not conducive to generating therapeutic agents that we need in today's situation and today's pandemic. So how do we address this problem? How do we get drugs that are effective out to the community, out to the populations where they're needed? Next slide, please. We take advantage of things that we already know. And the first thing that we know is that we have experience with developing antiviral therapies and interrupting the life cycles of viruses. Human immunodeficiency virus is a good example of approaches that we've used in the past to develop antiviral drugs. You see here the life cycle or the replication cycle of HIV, showing the essential elements of the replication cycle: entry into the cell, reverse transcription, integration, protease inhibitors, and the whole process of generating viruses that can be released from the cell to go out and infect other cells in the body. And knowing what we know about the replication cycle, we have identified critical points in the replication cycle that can be interrupted and that can be effective blocks of replication and the basis for effective drugs. So for example, reverse transcription is a critical step in the replication of HIV. And thus we have drugs that are effective against reverse transcriptase, the enzyme that mediates reverse transcription. We have other drugs that are effective and inhibit other processes within the viral life cycle that are effectively used in today's epidemic. Next slide. We can also take advantage of what we know about SARS, different coronaviruses. We have a novel coronavirus that we're dealing with in today's pandemic, but it's not the first coronavirus that we've had to deal with. SARS-CoV emerged in 2002. The MERS virus emerged for several years after that. And so we can take advantage of what we know about coronaviruses because the SARS-CoV-2 virus is one virus in a family of viruses. So we understand the genomic organization. We understand the structure of the virus. Next slide, please. And we also understand the replication cycle of coronaviruses in general, and specifically the SARS-CoV-2 virus. And knowing that, then we can identify critical points in the replication cycle that can be targeted for drug development and the development of therapeutic agents that will be effective against the virus. You can see examples of some of those drugs that are currently under development. And those drugs are specifically labeled to show their mechanisms of action and the points at which points within the replication cycle where these drugs interfere with the continued replication of the virus. Next slide, please. So what are we looking for in a good drug? Well, of course, we're looking for activity. We're looking for high antiviral activity against SARS-CoV-2. But we're also looking for safety. We're looking for selectivity. We're looking for a drug that targets the virus, but doesn't have any off-target effects that would create toxicity within the host. And we're also looking for drugs that don't have resistance potential. There's no potential for the virus to develop resistant mutations that may make that drug ineffective or less effective. Next slide, please. We do this by examining drug candidates in the laboratory, but then we also go into clinical trials. We take the most promising candidates and we put them through clinical trials to look at both safety and efficacy. Next slide, please. And this is done over a period of up to 5 or 6 years to get a candidate drug from the entry into the phase 1 trial to the end of the phase 3 trial and FDA approval. And this is all a process that involves looking first at safety in the phase 1 trial in a small number of healthy volunteers. This is an important part of understanding the safety of the drug, because this trial is a first look at different dosages, multiple doses of the drug to look for side effects that may pop up as those people are taking the drug. There's about a 30% failure rate at this point. The phase 2 trial then is the first look at efficacy, looking at safety again in a larger population of individuals, but looking at efficacy against the condition to which the drug is targeted. And of course, then this trial involves recruitment of individuals who are affected by the condition. In the case of COVID-19, this would be a trial that would involve people who are already infected with SARS-CoV-2. This process can take up to 2 years from the initiation of the clinical trial, and there's about a 66% failure rate. Then you get to phase 3 trials. These trials are the ultimate in demonstrating the efficacy of the drug and verifying the safety of the drug in populations that are much bigger and much more representative of the entire population of people who might be taking this drug. There are actually two trials that are required, 2 phase 3 trials that are required to validate the results that you get from the trial and to definitively say that this drug is an effective drug against the condition. And these trials can span 1 to 4 years because they're more involved, they're more complicated, they involve more volunteers, and the data analysis is more complicated. And this ends up with about a 75% failure rate. And so across all 3 phases then, you have about 1 candidate reaching final approval and FDA approval for every 17 compounds that enter clinical trials. Next slide. Once the last phase 3 trial is completed, that's where the FDA gets involved and they review the results of the trials and they approve, hopefully, the drug for clinical use and for use by physicians under the current circumstances. There may be a phase 4 trial that could look at other conditions or maybe even long-term effects of the drug. But once the drug is approved, it's ready to go out into the public. The good news is, and this is the last point at which we can take advantage of things we've already learned, next slide please, is that the drugs that are currently under evaluation for SARS-CoV-2 are drugs that, for the most part, have already been approved in clinical trials for other uses. So they've gone through safety trials, they've gone through efficacy trials for those other uses. And they're already being administered to patients for other reasons. So the process of bringing drugs on board for SARS-CoV-2 and for the current COVID-19 pandemic is going to be accelerated because some of those drugs are already out there. And one of my colleagues will be talking about these drugs in more detail in a presentation that's going to be following mine. Next slide, please. So where we're at right now is that we have The advantage of past research, SARS, MERS, these are all models for pathogenesis and the development of drugs that may be effective against SARS-CoV-2. We have drugs that can be repurposed or are positioned very well to be repurposed for the current pandemic. And there is some latitude available for accelerating clinical trials and eliminating some of the regulatory hurdles that that otherwise are in place for a more traditional and a more extended drug development process. But we also have to keep in mind at the very end here that we need to balance the expediency with safety. We want to get a drug that's effective against SARS-CoV-2, but we want to do that in a manner that's evidence-based, and we want to be making decisions that are good decisions that bring us to a drug that is not only effective, but also safe. Thank you for your attention, and I will now hand the presentation over to Dr. Kutzler.
Good afternoon. My name is Dr. Michele Kutzler. I'm an associate professor of medicine and microbiology and immunology at Drexel University. In this presentation, I will be covering the topic of COVID-19 vaccines. Next slide. As way of background, in regards to the epidemiology and clinical features of COVID-19 disease, it has been shown that common symptoms in patients with SARS-CoV-2 infection may be mostly asymptomatic, but often those infected present with fever, malaise, which is a general feeling of discomfort, myalgias, which include body aches, and cough. Approximately 15% of patients will develop severe disease and evidence of viral pneumonia. They may manifest by hypoxemia, which means that there's a below normal level of oxygen in their blood. It may also include multilobar ground glass opacities, which really means a partial filling of air in their lung spaces on chest CT scan. Also, severe cases may also show signs of dyspnea, which also means difficult and labored breathing. There also may be signs of coughing up blood. Next slide, please. As shown in the figure below, patients with severe disease may have complications. For example, many of these patients with severe disease will require ICU care, and they may progress to develop something called ARDS, which stands for Acute Respiratory Distress Syndrome. They may also show signs of shock and secondary organ failure and superinfections. Based on reports from China and preliminary estimates from the US, increased mortality from COVID-19 has been associated with older age and, importantly, underlying comorbidities. Some of these may include COPD, ischemic heart disease, diabetes, other underlying immunosuppressive disorders. But morbidity and mortality also may occur in patients who are younger than 65. But most of the mortality that's seen with this infection is in patients who are over 65. Next slide. An interesting finding is that SARS-CoV-2 can be transmitted from infected individuals who may not have symptoms. In addition, SARS-CoV-2 has caused pandemic disease within a period of weeks. So taken together, this suggests that in order to control this viral infection, it's going to be very challenging without the prospect of a vaccine. Next slide. I'd like to take a minute to review COVID-19 and the immune system and the role that antibodies play in protection. As shown in the figure on this slide, shown by the number 1, when a healthy person comes into contact with the virus for the first time, they usually don't have antibodies required to fight it. However, as shown in number 2, our body's defense systems kick in and we begin to produce antibodies against SARS. Our bodies then remember this immune response so that the person cannot be reinfected with the same virus. Vaccines function in the same way. When you're given a vaccine, your body recognizes that new antigen and begins to make antibodies against it. That way, your antibodies that you've developed against that antigen in the vaccine will protect you against further infections. Scientists can learn a lot from patients who become infected and survive infection. And what I mean by this is that their blood can reveal clues about the needed host immune response that leads to recovery and protection from SARS-CoV-2. So thus, vaccinologists study these immune responses to reveal clues about what is needed for protection against COVID-19 disease. Next slide. So in the case of COVID-19, once a person is exposed to SARS-CoV-2 virus, they should develop antibodies against the virus, and the presence of antibodies indicates immunity. Scientists are working to understand how long will this immunity last, and we predict, based on the SARS infections of early 2000s, that if you do develop antibodies against this SARS-CoV-2, it should last from 2 to 3 years. But scientists are working to further understand that question. Another question is that scientists are asking is whether antibodies may exacerbate the illness. And this may happen because sometimes a specific kind of antibody produced may actually activate other immune cells to produce cytokines that lead to increased disease. Recently, there were reports from Asia that patients who have recovered have subsequently tested positive again. The hypothesis or theory for this include that potentially there was a second infection. This is highly unlikely. Or it could be due to a chronic infection or perhaps a false negative test when the patient was actually still positive, which is more likely. So to get at the answers to these questions, scientists are looking at patients who recover from COVID disease, and what they're finding is that they have activated T cells and IgM and IgG antibodies that correlate with recovery. And so scientists and pharmaceutical companies working on vaccines are thus developing vaccine platforms that elicit both T and B cells. And these are the cells, B cells in particular, that make those important antibodies that we just talked about. Next slide. So the first step in vaccine development is to select immunogenic antigens to put into the vaccine. The SARS-CoV-2 virus uses ribonucleic acid as its genetic material. And SARS-CoV-2 seems to have a moderate rate of mutations to date, and this means that it actually should be susceptible to a vaccine. The SARS-CoV-2, as Dr. Navas-Martin talked about, is covered with spike proteins. And these spike proteins bind to a molecule on the surface of lung cells called the human angiotensin converting enzyme, or ACE2. There's a lot of spike protein on the outside of the virus, and this makes it a prime target for our immune responses. So most researchers have focused on the spike protein as an antigen for SARS-CoV-2. Next slide. There are several types of vaccine platforms that are used, and each kind has specific ways to deliver the SARS-CoV-2 antigen. So this slide and table summarizes current vaccines that are being developed that use something called nucleic acid vaccine-based products. The first column in this table shows the company name and the clinical trial number. The second column shows the type of antigen and how it's encoded and how it is delivered. There are several advantages to nucleic acid types of vaccines, and this includes rapid production, ease of design and manipulation, and the fact that these vaccines, once delivered to a patient, it leads to the induction of both B cell and T cell responses. A second positive advantage to these types of vaccines, particularly the mRNA types, is that they have the ability to mimic many aspects of natural viral infections. Thus, it improves stability and protein translation efficiency and induces robust immune responses. The disadvantages of this platform are shown. And below the table is a schematic which shows how nucleic acid vaccines are taken up by host immune cells, leading to the induction of both T cells and B cells. Next slide, please. This slide summarizes the current vaccines that are being developed that use viral vectors as a vehicle to deliver SARS-CoV-2 antigens as a vaccine product. There are several vaccines in the pipeline which are using the viral vector method to deliver the SARS-2 spike proteins. And they are using chimp adenoviruses or also Ad5 adenoviruses. And in some cases, they're using lentiviral vectors to transduce antigen-presenting cells, including dendritic cells. And then once these cells are transduced, they're using them to permit antigen processing and presentation of SARS antigens. And this results in activation of very durable, long-lasting T cells. The other advantage of the lentiviral vector system is it allows for multiple boosting, whereas the other adenoviral vaccine platforms usually only allows for a single immunization, because of some effects that happen with these types of these viral vectors, where you're not able to immunize a second time. And again, some more of those disadvantages are shown in the last column here. And then below the table is a schematic of how these viral vectors are taken up by host immune cells. The antigens are expressed on the surface of immune cells and presented to T and B cells, so that those cells can then make antibodies and other cytokines which are important against fighting off this pathogen. Next slide, please. The final table shows some other types of vaccines that are in the pipeline, live attenuated vaccines. The method in which the SARS-CoV-2 is presented to the immune system is that they use an attenuated virus, which is not pathogenic when used as a vaccine. But these are very effective, excellent in inducing T and B cell responses. And historically, these types of vaccines lead to lifelong immunity. So these are very interesting platforms that are being tested. Some of them are using bacteria as a carrier. And what's really exciting about this is the BacTRL, is that you can deliver this vaccine mucosally, thus inducing both mucosal and systemic immune responses. And since SARS-CoV-2 attacks the lung, having a mucosal immune response is very important. Below this table, again, is a schematic of how attenuated bacteria or attenuated viruses are taken up by the host immune response, leading to durable immune responses. Next slide, please. So I'd like to summarize with some key steps to developing COVID-19 vaccines. As I talked about, finding target antigens is the first step. Optimizing immunization routes, defining correlated immune protection. And from the data that's coming out of patients who have recovered from disease, we know that antibody responses are very important. In developing vaccines, testing in animal models is key. Scalability, building production facilities, creating a target product profile, and also working with epidemiologists to outbreak forecast and target populations who are most at risk and need a vaccine. Next slide, please. So remaining questions that scientists are working to understand more fully in order to develop an effective vaccine are, do people develop immunity? If humans do develop immunity, how long does it last, and will it prevent reinfection? What kind of immune response should vaccine developers look for? How do we know if a vaccine is likely to work? And we need a vaccine platform that will be safe. So I'd like to conclude by stating that this is really an exciting time for scientists in both industry and scientists working at universities such as Drexel University, research teams that are working to develop vaccines for prevention of SARS types of disease. So as we learn more about this virus and how long it will be in the environment, I'm really confident that the scientific community is working hard to develop effective vaccines, and that these vaccines will be able to protect us against reemergence of COVID. So as you will hear in the next talk, there are several potential therapeutics and vaccines in the pipeline. So I'd like to pass the presentation to our next speaker.
Thank you, Michele. Hi, everyone. I'm Paul McGonigle. I'm professor in the Department of Pharmacology and Physiology and a director of the Drug Discovery and Development Program at Drexel. As Michele just mentioned, the process of developing a vaccine can be long and complex, and we're unlikely to see approved vaccines for at least 12 to 18 months, if not longer. So I'm going to talk about therapeutics that are currently in kind of late stage development that have the potential to be available before vaccines are available. Next slide. First, I want to remind people of the potential benefit of antiviral drugs. One of the great successes of the pharmaceutical industry was the development of antiviral treatment for AIDS. So you can see in this chart that a year after antiviral drugs for AIDS were introduced in 1996, the mortality rate associated with AIDS declined by over 60% and continued to decline until at this day, back to the first slide, until could you go back to the previous slide? Until today, people who are infected with HIV it's no longer a death sentence. It is actually a chronic condition that people can live with. The drugs that are in late stage development for COVID-19 fall into two general classes. The first class is are antivirals that are designed to prevent either the entry or replication of the virus in host cells. And the second class of drugs are immunomodulatory drugs that are designed to address the hyperstimulation of the immune system that seems to be associated with COVID-19. Next slide. One of the drugs that's received a lot of publicity recently is a drug called remdesivir. Remdesivir is an adenosine analog that inhibits RNA-dependent RNA polymerase. In the figure on the right, you can see that RNA polymerase is responsible for copying strands of RNA that the virus needs to replicate. In step 2, you can see that remdesivir substitutes for adenosine in the RNA strand that's being synthesized. And in step 3, that you can see that synthesis is terminated. So the cell is no longer able to synthesize the RNA that's required for replication of the virus. Remdesivir was initially developed for the treatment of Ebola and Marburg's, but it was never approved by the FDA for those indications. Remdesivir has been shown to be active against multiple coronaviruses, MERS, SARS, and SARS-CoV-2, both in cell systems and in animal models. And even though it wasn't approved for Ebola, it was tested enough to be convinced that the compound is relatively safe and well tolerated. There have been a number of anecdotal reports of efficacy, and Gilead has scaled up their production of the compound, made it widely available for compassionate use, and have initiated a couple of relatively aggressive clinical trials to try to get this compound tested adequately and potentially on the market by the fall. Next. Another drug that's very similar to remdesivir is favipiravir, also known as Avigen. This is a purine analog that works by the same mechanism as remdesivir, not quite as selective as it substitutes for all purines. And there is some preclinical evidence that it can produce teratogenic or birth defects. There have been early reports in China that the drug is efficacious against COVID-19, and it's actually approved in China and Italy for the treatment of COVID-19, and there are additional trials that are currently underway. Next. Another drug that's received quite a bit of publicity is hydroxychloroquine and its analog chloroquine. These drugs have a relatively complex mechanism of action, as you can see in the panel on the right. They not only reduce protein trafficking within the cell, they also inhibit the glycosylation of the angiotensin converting enzyme that the virus needs to attach to in order to enter the host cell. In addition to that, these compounds have immunomodulatory effects. They have been in use for decades for the treatment of malaria. More recently, they've been approved for the treatment of rheumatoid arthritis and for lupus because of their immunomodulatory effects. And in general, at the doses used for those indications, they are generally well tolerated. However, it is well known that at high doses, they have cardiac toxicities and retinopathies associated with them. They've been shown, not surprisingly, based on the mechanism of action, they have been shown to be active against SARS-CoV-2 in cell systems, with hydroxychloroquine being more potent than chloroquine in these assays, and hydroxychloroquine in general being a little bit safer and less likely to produce retinopathies. The hydroxychloroquine has been approved for the treatment of COVID-19 in both China and Italy. However, there are recent reports of cardiac toxicity at high doses, a recent report out of Brazil, Just yesterday, there was a pre-publication report of a retrospective study of patients in Veterans Administration hospitals that demonstrated no benefit from hydroxychloroquine in relatively severe COVID-19 patients and a slight increase in mortality. Clinical trials are underway in Europe, in the US, Europe, and China, and there is hope that early intervention with hydroxychloroquine or use as a prophylactic might be more effective than how it has been used so far. Next. Another compound that works as a viral entry blocker is umifenavir or Arbidol. This targets the spike protein angiotensin converting enzyme interaction to block the fusion of the viral particle with the host cell. It's approved in Russia and China for the treatment of influenza and RSV, and has been shown to be active in vitro against several viruses, including SARS. There's preliminary data from China that it is effective, and it's a recommended option for treatment of COVID-19 in China. Next, please. Another class of antiviral drugs are protease inhibitors. Many protease inhibitors have been developed for the treatment of HIV. Lopinavir is a protease inhibitor that is selective for the 3 chymotrypsin-like protease. And on the right-hand side, you can see its mechanism of action, which is to block the processing of polypeptides and the creation of non-structural proteins that are required for replication of the virus. This compound has been approved for treatment of HIV, and it's used in combination with ritonavir, which is also a protease inhibitor, but weaker, and actually is used to prevent the metabolism of lopinavir. Lopinavir, this combination has been shown to be active against MERS in animal models. It also reduces the rates of acute respiratory distress syndrome in both SARS and MERS patients. However, one caution is that it is specifically designed to match a catalytic site that is present on the protease that is present in HIV, but this catalytic site is not present in SARS-CoV-2. Status is that there are preliminary reports indicating that it had little or no efficacy in the treatment of severe COVID-19, and clinical trials are ongoing. Next. So one of the consequences of COVID-19 seems to be an overactivation of the immune system. And one of the compounds that's being tested is a compound called tocilizumab, which is a monoclonal antibody that is an antagonist of the interleukin-6 receptor. This compound has been developed for the treatment of moderate to severe rheumatoid arthritis. And on the right, you can see that its mechanism of action is to block the interaction of interleukin-6 with its receptor, and thus block the downstream effects that are produced by stimulation of the IL-6 receptor. COVID-19 produces something that's called a cytokine storm. IL-6 is one of those cytokines that has been shown to be significantly elevated in COVID-19. Preliminary reports indicate that, there seems to be efficacy in using this compound in severe COVID-19, and there are multiple clinical trials underway to test this. Next. Similarly, saracatinib, also known as Kevzara, works by exactly the same mechanism. It is also a monoclonal antibody that's selective for the IL-6 receptor, and clinical trials are underway for this compound as well. Next. There are also immune system modulators that work by other mechanisms. One of those is interferon beta. Interferon beta is part of the innate immune system that Sonia mentioned earlier, and it interferes with viral replication and the spread by several different mechanisms. It has been shown to be effective in vitro and in vivo against both SARS and MERS. In general, it's combined with antivirals to help boost their effect, and in general considered safe and well tolerated. There are clinical trials that are currently underway in combination with lepanavir and ritonavir. Next. Another immune system modulator is baricitinib. So baricitinib is a Janus kinase inhibitor, and this inhibitor blocks the cytokine receptor signaling, intracellular signaling pathway to minimize or to modulate the immune response. However, it also blocks, by blocking these Janus kinase inhibitors, it also blocks the synthesis of interferon alpha, which may not be beneficial in the treatment of COVID-19. However, this particular compound also blocks something called AAK1 and AP2 associated kinase, which regulates endocytosis, which may prevent entry of virus into host cells. Clinical trials in partnership with NIH are underway with this compound. Next. Another approach, another class of immune system modulators are those that provide passive immunity. One of the approaches that has received a lot of attention lately is convalescent plasma. So this is a procedure that's been in place for almost a century, and it involves using the plasma from previously affected individuals and administer the antibodies that are produced by a patient infection can then be used to neutralize the virus in a patient with an active infection. Beneficial effects of this approach have been reported in the treatment of H1N1, Ebola, and SARS. And recently there have been anecdotal reports of benefit in COVID-19 patients. Obviously, one of the limitations of this is it's going to be hard to scale that up. However, using the same general rationale, the biotech companies Regeneron and AbCellera are trying also to develop therapeutic antibodies. Regeneron has used mice with a genetically modified immune system to produce multiple human antibodies. And they are screening those antibodies with the goal of identifying a cocktail of 2 antibodies that'll be selected on the basis of their potency for the SARS-CoV-2 spike protein. And their plan is to initiate clinical trials early this summer. Similarly, AbCellera has used their rapid pandemic response platform to screen millions of immune cells for neutralizing antibodies. They've identified over 500 unique human antibodies for SARS-CoV-2 within a week of receiving blood from one of the first patients in the US who recovered from the viral infection. They also plan to initiate trials later this summer. Next. So in conclusion, remdesivir and the antiviral drugs, remdesivir and favipiravir, have shown early promise. Hydroxychloroquine has been somewhat disappointing to date. However, there's still a lot to be learned about whether early treatment can show some efficacy. The immune modulators tocilizumab and saralimab have shown early promise to minimize symptoms. Convalescent plasma also shows promise, but it'd be very difficult to scale up. But I should point out that this is just the tip of the iceberg. There are currently 48 compounds that are in clinical trials and 207 development programs currently underway for the treatment of COVID-19. And I'd like to take this opportunity to personally thank all of the scientists who are working at pharmaceutical companies, biotech companies, academic laboratories, and government facilities to develop therapeutics for COVID-19. And finally, next. The bottom line is that we need more data from well-controlled clinical trials to identify which of these therapeutics are truly effective. And Dr. Rich Mangano is going to describe the status of those trials and some of the challenges associated with them.
Thank you, Paul. Hello, everyone. I am Richard Mangano. I am the director of the graduate programs in clinical research at Drexel University. If we can start with the next slide. As you've seen in Dr. Krebs' presentation, a typical timeline for the development of a new therapeutic can take from 10 to 15 years from initiation of discovery research through regulatory approval and product launch. This development model is obviously ill-suited when a novel pathogen results in a pandemic that affects an exceptionally high proportion of the global population. Compressing this timeline involves a number of trade-offs with multiple activities being conducted in parallel and with substantial financial risk to drug developers. You can see in this figure that manufacturing development starts at the same time as clinical development without knowing if a vaccine or a novel antiviral will prove to be both safe and effective in preventing SARS-CoV-2 infection or preventing the development of acute respiratory distress syndrome in those who are already affected. Next slide. Several global regulatory agencies have provided guidance to aid in the clinical development of vaccines and antiviral compounds. The FDA has developed 3 documents that are relevant in particular during this pandemic. The Center for Biologics Evaluation and Research issued a guidance in 2007 addressing the clinical data required for the approval of pandemic flu vaccines. The Center for Drug Evaluation and Research issued a guidance in 2011 discussing the development of antiviral drugs for the treatment or prophylaxis of influenza. One thing I could point out in this particular guidance is that there are different clinical endpoints for hospitalized patients and for demonstrating disease prevention in those who haven't been infected. A very timely draft guidance for demonstrating substantial evidence for effectiveness for human drug and biological products was issued jointly by both CDER and CBER just last December. It's out for public review and comment through the FDA at this moment. This guidance is particularly relevant in our current situation. The FDA and drug developers are bound by a statutory standard requiring substantial evidence for the approval of a therapy as safe and effective. In normal development programs, this is interpreted as the requirement for at least 2 positive phase 3 studies. However, the approaches discussed in this guidance can yield evidence that meets this statutory requirement with only 1 positive phase 3 study. This should help greatly in speeding along drug development for this particular pandemic. Next slide. Health and Human Services became concerned during the 2009 swine flu pandemic that there were no approved antiviral drugs for hospitalized patients. There were many antiviral clinical trials during this pandemic, but none of them were successful in demonstrating efficacy. As a result, several HHS agencies formed an interagency clinical endpoints working group. This working group has identified several factors that might improve hospital-based therapeutic trials. These included primary clinical endpoints, increased clinical site enrollment, and appropriate baseline enrollment criteria. The major obstacle identified by the working group was a lack of clinically meaningful, validated primary endpoints. Their recent recommendation was the use of a 6-category ordinal outcome in these trials. Those categories include death, ICU admission with or without mechanical ventilation, hospital ward admission with or without supplemental oxygen, or patients not hospitalized at all. Validation of these endpoints is ongoing in several non-COVID-19 influenza studies. Next slide, please. With this background, let's take a look at the current clinical trial landscape. The good news is there are a lot of trials underway, and unfortunately, the bad news is there are a lot of clinical trials underway. The data on this slide were derived from the clinicaltrials.gov website last Sunday evening. I returned to their website this morning, and now the total number of registered studies is 795. The table on the right shows the breakout by study type: interventional, observational, and expanded access. Searching specifically last Sunday with the terms antiviral and vaccine resulted in 45 and 11 studies respectively. There are likely more COVID-19 studies ongoing globally that aren't included in this registry. These trials can be found in the European Union and other national trial registries. Next slide. One very large international study ongoing is the Solidarity trial launched by the World Health Organization. As of the last update on the WHO website, this trial was being conducted in over 90 nations at 960 sites. Solidarity is an open label study in which the study participants know the treatments to which the patients are randomly allocated. These 5 treatments include local standard of care, or the local standard of care plus one of the 4 treatments listed below: remdesivir, chloroquine or hydrochloroquine, lopinavir with retinovar plus interferon beta 1A. There are planned interim trial analyses in this trial that are monitored by a global data and safety monitoring committee. This committee can drop arms that are ineffective and add arms if it's warranted when new treatments prove to be effective. Information that's collected when the patient is discharged or dies is which study drugs were given and for how long, whether ventilation or intensive care was received, and if so, when it began, date of discharge, or the date and cause of death while still in the hospital. The projected study end date for this study is March of 2021. Next slide. Several of the more prominent antivirals being tested in COVID-19 are listed in this table. If you want to dig into the specifics of the trial, the trial identifier is provided here and can simply plug it in and search for the details. Gilead's remdesivir is not an approved drug at this point, but as Paul stated, has been studied in Ebola and other infections. Actemra has been approved for treating rheumatoid arthritis. Kaletra is approved for the treatment and prevention of HIV/AIDS. The Kevzara is also approved for treating RA, and hydrochloroquine is used to prevent or treat malaria. These last 4 drugs are examples of therapeutics approved for other indications that are being repurposed for the treatment of COVID-19. All of these drugs are in phase 3 studies. Gilead's 2 studies in patients with severe symptoms and second study in patients with moderate symptoms have aggressive timelines. These studies started in March and they're estimated to conclude in May of this year. The Kaletra study is scheduled to complete in November, while the remaining studies run through next year and end in either the first or third quarter of 2021. The duration of these studies is largely a function of the study design. Next slide. All of these antiviral studies, patients are randomly allocated into parallel treatment groups. Remdesivir studies are open label, while the treatment in all the other studies is blinded. The control group in the remdesivir studies is a standard of care, while the remaining studies are placebo-controlled and should yield valuable information. Several of these studies are using a 7-category ordinal scale as the primary outcome, while the Kaletra prevention study is looking at the rate of SARS-CoV-2 infection, and hydrochloroquine is looking at all-cause mortality in that particular study. Should be noted that earlier this week, Novartis won FDA approval to initiate a US study with hydroxychloroquine as well, although study details aren't available at this point. On the next slide, we can switch to the vaccine studies that have been initiated. And this month, all but one of these vaccines are novel and in phase 1 studies in healthy volunteers. The primary outcome in these phase 1 studies are safety related, and these subjects are being followed for 6 to 12 months. Secondary outcomes include the increase in IgG titer from baseline, antibody titer, and the percentage of subjects who seroconvert, and that is more routinely being defined as a 4-fold increase in antibody titer in these healthy volunteers. Moderna was the first company to initiate a study in the US in partnership with the National Institute of Allergy and Infectious Diseases. This study is scheduled to conclude in June 2021 because the subjects are being followed for safety for 1 year. Inovio's study began this month, and they're following subjects for only 28 weeks, and that results in the shorter study duration. The one phase 3 trial for a vaccine is for a vaccine already approved to prevent tuberculosis. This is being conducted in Australia and will recruit over 4,000 healthcare workers. We hear a lot about having a vaccine in 18 to 24 months, and the validity of a statement like this is going to depend on one's definition of having. I think the more pertinent questions we should be asking in these trials is how long will it take to identify a potentially viable vaccine candidate? When will confirmatory studies start? And how long do those studies have to be conducted in order to approve the vaccine as safe and effective before we start treating hundreds of millions of people globally? Finally, on my last slide, we can skip one more. This presentation is certainly not an exhaustive overview of an extremely large and challenging global endeavor. What I'd like to do here is summarize a few of the challenges that we're facing in conducting these trials. And first is the social challenge of learning, conducting clinical research versus doing, as in treating patients. For ethical reasons, medicine is organized to perform each task separately, and we're now struggling to combine them to gain as much information as we can. The large and growing daily number of studies are competing for patients and a limited number of resources. Trained personnel, protective equipment, required tests and assays are challenging. Many of these studies are unblinded. Some have no control groups. Many don't have a placebo control group. This will challenge us in interpreting the study results. There are also inconsistent study endpoints. Trials of novel vaccines are starting now with minimal and no preclinical safety data. These preclinical studies are running in parallel with the phase 1 human studies, and this could pose an increased risk to volunteers in the phase 1 studies. So while Paul was rightly giving props to all the scientists involved with this, let me speak on patients and volunteers' behalf and thank them for taking part in these studies. The determination of vaccine efficacy may be confounded by preventive practices that we're all involved with now. Is a vaccine really working, or are we seeing that quarantine, isolation, social distancing, wearing of face masks is actually preventing the spread or contracting the infection? And equally important, clinical trials for all the other equally serious diseases that have been conducted are at a virtual standstill at this point. Few planned studies are being initiated, if any, and patient study visits in trials that were underway have been shifted to virtual visits when possible, but this has an impact on assessment of safety data in these studies here. So with that, I'll stop here, and speaking for my Drexel colleagues, we thank you for your interest in this webinar.
Thank you so much. At this time, we're open for questions. Just as a reminder, we will try to get to everyone's questions as best we can. If your question was not answered in today's presentation, it will present itself in a question and answer webpage post-webinar. We will provide a link via email after the page is updated. Stephanie?
Hello all. I have communicated with you off and on and conveyed some of your questions to the speakers. Dr. McGonigle and Dr. Krebs, Dr. Kutzler, I have asked via chat for you to maybe address some of the questions. For example, Dr. Kutzler, there is a question, could plasma donation for antibody transfer reduce the immunity of the donor?
Yes, that's a great question. Thank you, Stephanie, and thank you to the attendee who asked that question. So when serum donations are given and antibodies purified and used as immune therapy, donors do not lose the ability to continue to make that antibody. So you have memory B cells that are differentiated into plasma cells, which make antibodies. So even if serum is donated, you still have those B cells remaining that will continue to make that antibody in the donor. So there is no loss of immunity in the donor.
Thank you, Dr. Kutzler. To Dr. Krebs, maybe, has there been any indication of patients who are on current HIV therapy, you mentioned, not becoming infected with COVID or having mild symptoms? Dr. Krebs?
I can take that as well, Steph.
Okay.
This is Dr. Kutzler. So I think that's a really interesting question. The hypothesis is that the antiretroviral treatment that HIV infected patients are taking may protect them from SARS-related diseases. However, there is not enough data. I think Dr. Navas-Martin would agree with me on this. There's not enough data or large enough studies to really show that that the disease of SARS is different in HIV positive patients versus negative. So right now, there is not enough evidence to suggest that SARS is less than HIV infected, although it's a very interesting question.
Sorry, I wasn't fast enough on my mute button. I would agree that the questions regard the risk of infection as well as the effects of HIV infection, concurrent infection on pathogenesis. And there isn't enough information out there to make positive statements about the interaction between SARS-CoV-2 and HIV.
Thank you. And Dr. Navas-Martin, you addressed the question, what are the differences between SARS virus from the early 2000s and SARS-CoV-2, and how would it theoretically affect the immunity period? Do you want to address this verbally, or do you want me to just review your responses?
No, I can address this verbally. The two viruses are different, although they have the same origin, and this has been demonstrated by phylogenetic analysis, by sequencing and analyzing with particular softwares and programs, many sequences originated from coronaviruses isolated from bats. And also comparing those sequences with the known sequences for the 2003 SARS coronavirus and the 2019 SARS coronavirus. So clearly these two viruses are different, but clearly they both were originated from circulating coronavirus in bat populations. And whether the immunity against these two viruses is similar or not is a question for our current work. And I don't have a clear answer to that. But clearly the data that I show in my presentation from the German group that published a Cell paper clearly demonstrate that at least some neutralizing antibodies, maybe not all, but some, can neutralize both 2003 SARS and 2019 SARS coronavirus.
Thank you. And maybe we can take one more for Dr. McGonigle. What was the rationale for approval in China and Italy for the various therapeutics approved there? When speaking of Italy, it's part of the EU, meaning it should be under the EU approval rules. How is it approved in Italy, but not in all of the EU? And is it some sort of compassionate use of emergency approval only in Italy?
So those are both very good questions. I'm not certain of the regulatory process that exists in China, but I'm assuming that because they had so much early experience that they are privy to data, and they made their decision based on the data they were seeing in the early administration of those drugs. With regard to Italy and approval of drugs, first, although the approval system is coordinated in the European Union, each country has its own final approval and approval list of drugs, so that pharmaceutical companies need to get approval from individual countries, not just the European Union. So in the case of Italy, they had the ability to approve that drug for the treatment. And I suspect that because of the urgency, that they were fairly liberal in terms of their criterion for approving that drug.
Thank you, Dr. McGonigle. And I'll take one more. Rena, you stop me when we're out of time, please. There is a question from maybe Dr. Kutzler or Dr. Navas-Martin. Do we know, do you have an opinion about the correlation of ACE2 levels in patients with diabetes or taking anti-hypersensitive drugs and the mortality of COVID?
I think we don't know the answer to that question. It's a very important point. And indeed, there is some mortality in these kind of patients. Why not? Maybe ACE2 has potentially a pathogenic role in the disease, but I don't have a clear answer molecularly how it may happen. But the potential is there, of course.
Yes, and I agree. I echo Dr. Navas-Martin's response. I don't have anything to add. Thank you, Dr. Navas-Martin.
Thank you. All right. Dr. Navas-Martin also responded to the question of long-term effects. Do you want to elaborate, Dr. Navas-Martin?
Yes, sure. We currently don't know long-term effects, but one thing is true is that if we take back and take a look to the 2003-2004 epidemic in China, those survivors, I haven't really followed, I have followed the literature, but I haven't really identified major detrimental effects of SARS coronavirus infection on those 2003 survivors. So with this new virus, COVID-2, it's true that seems like there is also a multi-organ component and even the central nervous system could be involved. So it's something for the near future and current investigations on how this virus may impact other organs other than the lung. So the long-term effects, the potential long-term effects of COVID-2 needs to be further investigated.
All right, I have two more questions. One, are people on ACE inhibitors or ARBs protected from COVID-19 to a degree or at all?
That's for me, the question?
Whoever wants to take it, I think you would probably be the right person to talk about it, but if someone else has a comment on it, please feel free.
I mean, I don't think there is clear data on that. I don't think there is protection because you take the inhibitor. And I have to also explain that, you know, just the expression of ACE2 does not really correlate completely with infection. And in some circumstances, maybe there is also the potential that the virus use other molecules to enter the cell.
Good point. Dr. Mangano, do you think a 28-week clinical trial for vaccine ending in November 2020 before the flu season would suffice when others go a year or more?
Mm-hmm. It can. I think, you know, what they will have to do, those patients, if they come down with adverse events beyond that 28-day window, would be instructed to report in anyway. So, you know, this, I think, primarily is to show that you can safely administer the vaccine without any serious untoward effects for it. And that would obviously lead into a larger phase 3 study in which patients or the population would be followed for at least a year.
Thank you.
I think for regulatory agencies is when they feel they would have enough safety data to give them confidence to approving, releasing the drug.
Excellent. And if someone is on a drug that reduces B cells, how effective might a vaccine be? Maybe Dr. Kutzler?
Yes, I think in order to, the most important immune correlate of protection is anti-SARS antibodies. So if you have a patient who is on some type of therapy for a comorbid condition where they lack B cells or their B cell function is suppressed, then it will be very difficult for them to develop antibodies. So in those cases, the prophylactic immune therapy would be much more effective, where you're giving them passive transfer of immunoglobulin. But so there are all alternatives for those types of patients.
Thanks, Dr. Kutzler. And maybe Dr. McGonigle, the question is, can any of the panelists contribute to the question of addressing the possibility of medical devices regulating the cytokine storm and increased inflammatory response syndrome as an adjunct therapy, specifically relating to NETs?
I'm not familiar with medical devices. Rich, did you hear anything about those?
A study, I believe, had been approved for a device that is removing the cytokines from circulating plasma in infected patients, hospitalized patients. That's about all the detail I have on that study at this point.
Yeah, maybe it sounds like it might be a little bit involved. Maybe this is something we can address in the Q&A webpage. And thank you very much for the question. Reina, where are we on time?
We have 5 more minutes.
All right, perfect. Then we have at least one more question. Dr. Krebs, or someone else, can multiple different coronavirus infections occur simultaneously, or will established infection with one coronavirus preclude an additional infection in the same individual? And if yes, are there any examples or explanations?
I don't think there's enough known about the cross-reactivity between SARS-1, MERS, and SARS-CoV-2 to comment on how one infection would affect another. Perhaps Dr. Kutzler could comment on the potential crossover of immunity to those viruses.
So Fred, could you repeat your question? I missed the question.
I think the question has to do with concurrent infections, and particularly
Crossover immunity.
2 different coronaviruses.
Yes, I think these coronaviruses are from different, they originate from different viruses. So I think if you have antibodies to one type of coronavirus, it doesn't protect you from another. And that's why with colds, you can get colds over and over again. That's why, any vaccines that were developed against the first coronavirus in 2003, they would not be effective against this coronavirus. So the viruses are different, their envelope on their surface, so antibodies would not be cross-protective.
Thank you very much. Do we have time for one more? I think we've eaten up our 5 minutes. Reina?
Sure, we can conclude at this time. Excellent.
Thank you very much for all of your questions. As Reina and I mentioned before, we will aggregate the questions which are manifold, very in-depth, and very important, and address them on a FAQ page, which we will post and then distribute to all of the registrants.
Yes. Thank you again for joining us this afternoon, and a special thank you to our distinguished panel of faculty. for taking their time to be with us and share their expertise. Again, this webinar was recorded and will be sent in a follow-up email. Have a great afternoon, everyone.
Thank you. Bye-bye.